Hey guys, Mr. P. In this video we're going to talk specifically about anaerobic respiration, which means what happens when oxygen isn't available. We are aerobic organisms, and so far in this series we've talked about what happens when oxygen is present, meaning the entire aerobic cell respiration pathway. We talked through the intricacies of glycolysis, prep step, Krebs, and chemiosmosis. When oxygen is present, oxygen acts as that final electron acceptor, which means it accepts those high-energy electrons as they come through the electron transport chain, producing water by grabbing a few hydrogen ions, and eventually leaving the system. Now, we are going to kind of switch focus and talk about what happens in the absence of oxygen. And so, what is fermentation? Well, there are two types of fermentation that we need to know about, one being lactic acid fermentation and the other being alcoholic fermentation, both of which have incredibly important applications, not only in the area of biology, but in the area of human physiology or in the food and beverage industry. Lactic acid fermentation is the type of fermentation that humans and other animals do within their muscle cells. Now, if you've ever gone running or swimming or weightlifting or jogging or doing anything strenuous, simply moving things around your room or moving things around your house, moving furniture, that kind of thing, that often leads to muscle soreness, and that's because in your muscle cells, which are living tissue cells that utilize a lot of ATP, they require a lot of oxygen, and as that oxygen level depletes or starts becoming depleted, your muscle cells switch from aerobic respiration to anaerobic respiration in an effort to keep you alive and to produce a little bit of ATP, and we'll talk about that in a minute. Now, alcoholic fermentation would be within yeast cells, which is obviously one of the applications and one of the ingredients for baking bread. And again, we'll talk about that in a minute. Okay, so yeast cells have to be added to dough in order to cause it to rise, and the yeast cells are going to be producing the alcohol, which is produced during the dough rising process, which will eventually get baked out of our bread, and that leaves these characteristic holes that we become used to seeing within a loaf of bread. Muscle cells are going to go through lactic acid fermentation. Yeast cells are going to go through alcoholic fermentation. What is anaerobic respiration? We've kind of alluded to that already, but anaerobic respiration is when O2 or oxygen is not present. So we are aerobic organisms, meaning we require oxygen to live. However, we're not always in an aerobic state. We as an organism require oxygen, but whether or not that oxygen is fully diffused into our muscle cells is what we're really talking about. We can have high oxygen stores, we can have low oxygen stores. The more strenuous activity we do, like lifting weights, aerobic calisthenic type activities, and then sprinting or swimming, anything that builds muscular strength, muscular power, muscular size, connective tissue strength, or high intensity endurance, anything that is centered around those goals is going to be an anaerobic exercise. Anaerobic exercise burns through the stored O2 levels in your muscles quickly, leading to oxygen depletion, which obviously means oxygen is no longer available to be used within that cellular respiration pathway, leading to anaerobic respiration, which is going to be talked about in a minute. We, again, humans, are aerobic organisms, but do go through anaerobic respiration at times when oxygen levels are not full. We go through lactic acid fermentation, and so this is the lactic acid fermentation pathway. You will notice some familiarities, but then you'll obviously notice some differences. You know at this point glycolysis. Remember, glycolysis takes glucose, which is the same glucose we've talked about, C6H12O6. It's that same 6-carbon compound, C6H12O6, which is oxidized and split into 2 pyruvates. Remember, the pyruvates are the 3-carbon compounds, which are made at the end of glycolysis. Obviously, this part of the pathway is glycolysis. We take a glucose, we oxidize it, we take 2 molecules of NAD+, We turn them into 2 molecules of NADH. We've always said that NADH is our full taxi. They grab 2 electrons and a hydrogen ion each. In the process, we have a net gain of 2 ATPs, ATP being our high energy compound. We produce those 2 net ATPs by taking ADP, adding a phosphate from our fructose 1,6-bisphosphate. Remember, we had to invest 2 ATPs in order to produce 4. That leaves us with a net of 2. These ATPs are going to be used by the cell. This is really important. We'll come back to that in a minute. And in the process of producing these 2 net ATPs and these 2 NADHs, we produce the 2 pyruvates, which exit glycolysis. Now, there's kind of a fork in the road at this point. If oxygen is available within the muscle cells and therefore utilized by the mitochondria, these pyruvates will enter the prep step, which turns that pyruvate into that 2-carbon CoA, which remember is called acetyl-CoA. That's if oxygen is present. However, if oxygen isn't present, it shuts down that cell respiration pathway, and it requires us to either sit on a whole bunch of pyruvates, because remember, glycolysis is an anaerobic process, meaning glycolysis will happen regardless of whether oxygen is available or not. And we know that it's an anaerobic process because when we look through the metabolites that are produced and the intermediate processes that are happening within this glycolysis pathway, there is no oxygen being used anywhere within the steps of that glycolytic pathway. Once our pyruvates are made and it is determined that oxygen is not available, it shuts down the cell respiration pathway, meaning we cannot produce acetyl-CoA, we cannot start the Krebs, we cannot go through the ETC, we cannot do chemiosmosis, we therefore can't produce a whole bunch of energy, and we either, as I said before, have to sit on a bunch of this pyruvate, or we can ensure that the glycolysis pathway continues to give us a little bit of energy to be used right now to keep ourselves alive by simply taking these NADHs, which cannot go to the ETC, which remember is where they want to go. They cannot go to the ETC because the entire cell respiration pathway is shut down. So these NADHs are going to simply give the pyruvates back their electrons and hydrogen ions, converting them back into NAD+, turning the 2 pyruvates into 2 lactates, which is another word or another name for lactic acid. Again, producing 2 of those. So the entire lactic acid fermentation is taking glucose, producing 2 pyruvates, going through the normal glycolytic pathway, producing 2 ATPs net per glucose, just to then take the pyruvates, turn them to lactic acid, and that is incredibly important because it frees up our taxis, NAD+, to return back to NAD+ within the glycolytic pathway to ensure that we can continue to break down glucose for the sole purpose of producing a net profit of 2 ATPs. If we did not keep this process going by converting NADH back into NAD+, we could not sustain this net ATP production per glucose to produce a little bit of energy to keep ourselves alive. So this lactic acid fermentation process for us is all about life sustainability. This is not a process by which we produce an ample amount of ATP. It is simply a way for us to produce a little bit of energy, which is better than nothing, which helps to keep ourselves alive. So if we bring this back to our whole picture, we've seen this in previous videos. Again, remember, glycolysis is an anaerobic process, meaning it will happen with or without oxygen. This is going to happen every single time. Glucose is turned into 2 molecules of pyruvate. When oxygen is present, the NADH that is made during glycolysis and the pyruvate, which will eventually be shuttled through the prep step and turned into acetyl-CoA, will enter the Krebs, ultimately to produce more of this NADH and FADH2 for the sole purpose of shuttling all of those high energy electron carriers to the ETC for chemiosmotic phosphorylation. Now, the purpose of the Krebs, and most importantly this chemiosmotic phosphorylation, is to produce a whole bunch of ATP. This is obviously the purpose of aerobic respiration. However, like I said in the last slide, when oxygen isn't present, it shuts down the shuttling of that NADH because it simply shuts down this final electron acceptor. It therefore shuts down chemiosmosis, it shuts down the citric acid cycle, and it shuts down the prep step. The NADH that was made during glycolysis and the pyruvate that is made during glycolysis will be made, and again, we do produce a little bit of bonus ATP during glycolysis. Remember, we have a net of 2 ATPs made. That will continue to happen. However, pyruvate will not be shuttled into the prep step and therefore enter the Krebs, and NADH will not be shuttled to the ETC. Instead, that NADH will head to the anaerobic respiration pathway, which for us, in a human's case, will produce lactic acid, but if this was a yeast cell, this NADH and this same pathway would produce ethanol. NADH is going to give the 2 electrons and hydrogen ions back to the pyruvate, turning that pyruvate into lactic acid or lactate so that we can ensure that this glycolytic pathway continues to produce a little bit of bonus ATP to keep ourselves alive. Again, that's a life preservation pathway. It keeps us alive when oxygen isn't available. That's it for this particular video. I hope you learned something. If you have a question, leave it in the comments. Subscribe to the channel. We will see you later.